Hello, and welcome to this demo of PlotMicrobiome, a user-friendly tool for microbiome sequencing data analysis. PlotMicrobiome provides an intuitive interface and clear navigation. You do not need any coding experience to use it. The pipeline is also highly reproducible. If you run the same analysis again with the same options, you will get the same results every time. In this demo, I will walk you through the major functions of PlotMicrobiome, show how to generate different results and figures, and demonstrate the AI-powered analysis report that can help you quickly exploit your dataset. First, let's look at the data input module. PlotMicrobiome accepts both amplicon sequencing data with taxonomic structure and shotgun metagenomic data with taxonomic structure. In this cases, your abundance table already includes a final column with a taxonomic ID. The column contains the full taxonomy for each feature or ASV, for example, phylum, class, order, and all the way to genus and species. You can also use tables without taxonomic structure. This can be any kind of feature table, such as functional pathway abundance or gene abundance tables. The app accepts comma-separated values, tab-delimited text files, and biome files. For amplicon sequencing data, these are typically direct outputs from tools like KIME2 and Data2. For shotgun metagenomic sequencing data, they're often direct outputs from tools such as Kraken2 and Metaflon. You can upload these files directly without manual formatting. After you upload the files, you can set a few basic options. You can specify whether the table is in biome format, whether it's an ASV table, and what delimiter it uses, for example, comma or tab. You can also exclude samples with very low read counts. For example, if a sample has only a few hundred reads, it may be poor quality and not suitable for downstream analysis. Here, you can set a minimum read threshold, and samples below that cutoff will be removed. The next step is data normalization to correct for differences in sequencing depth across samples. In most microbiome studies, each sample has a different number of reads. PlotMicrobiome supports several normalization methods. You can scale counts based on proportions and the average sequencing depth, or you can perform rarefaction. If you choose rarefaction, you specify the target number of reads, and each sample is then rarefied to that depth so that samples are more comparable. For this demo, I will quickly load an example dataset so we can focus on the features of the app. The example dataset is 16S amplicon sequencing data from 40 participants living in rural and urban China. The reference to the original study is shown here, and the data are publicly available in the NCBI bioproject listed in the publication. Once we select the example dataset, we click Run. The app then shows a preview table where columns are samples and rows are taxonomic IDs or ASVs. By default, only a few rows are displayed, but you can increase this to show more rows or more columns. You can also download the R code used for processing so you can see exactly which functions were run. The normalized abundance table can be downloaded as well. The app also shows how many rows and columns are in the dataset, so you know the number of features and samples. Next, we go to the metadata input module. Here you upload your sample metadata file. This can also be a comma-separated or tab-delimited file. You choose which column contains the sample IDs, and those IDs must match the sample names in the data input table. In this demo, I will again load the example metadata. After clicking Run, the app confirms that there are 40 samples and that the sample names match between the data table and the metadata table. Now we are ready to use the AI-powered analysis report. This feature is built on open source large language models. In the future, we may also add paid models for more advanced or faster analysis, but currently all available options are open source. Different models may take different amounts of time to run. Here you can choose which AI model to use. Available options currently include DeepSeek, Gemma, Mistral, Qwen, Llama, and GPT-OSS. In this demo, I will select Mistral. Next, we select a metadata variable for the main comparison. Here we use rural versus urban status. You can also provide a short background description of your dataset. This helps the AI model better understand the context and interpret the results. For example, we can write, these are gut microbiome samples from rural and urban residents in China. Once the model, metadata, and optionally background text are set, we click Generate Report. The app will run a series of analyses and then ask the AI model to summarize the results. Depending on the model and dataset size, this may take a few minutes. So in the demo, we will fast forward. When the report is ready, you can download it as an HTML file. You can open this report in any web browser, such as Chrome, Safari, or Firefox. The report includes the name of the AI model used. It's important to remember that AI models can make mistakes, so all interpretations and conclusions should be reviewed before drawing scientific inferences or including them in publications. Let's take a brief look at what appears in the AI report. First, it shows a PCOA analysis of beta diversity by rural versus urban status. In the example, rural and urban samples are separated on the ordination plot. Ellipses around each group represent 95% confidence intervals. In the title, we see the Permanova result. For example, the R-squared might be around 12% with a p-value of 0.002, indicating a significant difference in community composition between rural and urban groups. The report includes multiple PCOA axes, such as axes 1 and 2, 3 and 4, and so on. The report also shows box plots of the PCOA axis scores by group, illustrating how rural and urban samples differ along each axis. Underneath, the AI generates a summary of the PCOA results, describing which axes show stronger separation and how rural and urban communities differ. Next, the report includes alpha diversity analyses. In this example, the alpha diversity of rural and urban samples is not significantly different. Most p-values are higher than 0.05, and the AI summary explains that alpha diversity is similar between groups. Then the report shows taxonomic composition at different levels, such as phylum and genus. There's a simple average composition plot for each group, and the AI summarizes which taxa are more common in rural versus urban participants. The report also includes differential abundance analysis. It lists the top significant taxa based on tests such as the Wilcoxon-Rank sum test, along with the test statistic, p-values, FDR-adjusted p-values, the group in which each taxon is enriched, and the fold change. There are box plots for the most significant taxa. For example, some taxa, such as Corydobacteriaceae, may appear much higher in rural than in urban samples. The AI then provides a narrative summary of the differential taxa results. Finally, the report includes an AI-generated discussion that summarizes the main findings across all analyses. We are still expanding this feature to include more plots and results in the AI report, but the existing version already demonstrates how quickly you can obtain a structured summary for your dataset. You can download the report at any time. Because it is an HTML file, you can open it easily on any computer and share it with collaborators. After you review the AI report, you may want to explore specific analyses in more detail using the other tabs. These provide more flexibility and control over the figures and models. Let's look at the ordination and MDS plot module. Here you can reproduce analyses similar to the PCOA in the AI report, but with more options. You select the metadata variable for coloring or testing, choose the taxonomic level such as genus, and choose the ordination methods such as PCOA or non-metric MDS. You can select the distance measure, decide whether to normalize the data, and adjust plotting options. These include point color, size, transparency, label size, and legend position. After clicking Run, the ordination plot appears. You can make the points larger and less transparent, enlarge the sample labels, move the legend, and even label individual sample names if you want to inspect specific samples. You can also make the overall figure larger to improve readability. You can also analyze subsets of the data. For example, you can restrict the analysis to only female participants and then rerun the ordination. This allows you to investigate whether rural versus urban differences are consistent within subgroups. Next, we have the alpha diversity module. Here you can again choose to analyze the full dataset or a subset based on metadata, such as only females or only a specific age range. You select the test variable, and then you can calculate alpha diversity at different taxonomic levels, for example, from phylum to genus, species, or AFV level. The app generates box plots for each group. You have more flexibility to control the display, including the order of groups. For instance, if you prefer to show urban on the left and rural on the right, you can change the order and rerun the plot. This module is designed to help you produce publication-ready alpha diversity plots with minimal manual editing. The next module is the bar plot visualization of taxonomic composition. The concept is similar. You choose a taxonomic level and a grouping variable. You can plot the composition for all samples individually or aggregate by group. In many cases, it's informative to plot all samples and look at their individual compositions. To do that, you can select all sample IDs so that each bar represents a single sample. You can change the orientation to vertical bars and increase the font size. This creates a straightforward visualization of how taxonomic composition varies across all samples in the dataset. Now we move to the first step of differential abundance analysis, filtering taxa. Microbiome datasets often contain thousands of taxa, many of which are very low abundance or present in only a few samples. If you include all of these in statistical tests, you may get spurious results and reduced power. In the filtering module, you can select a stratification variable if you only want to analyze a subset. For example, only female participants. You can choose whether to include or exclude ASV level features, and you can set prevalence and abundance cutoffs to remove very rare taxa. After you click Run, the app reports how many taxa remain after filtering. Those filtered taxa are then used in subsequent statistical analyses. Once the filtering is done, we move on to the core step for finding differential taxa. Here we select rural versus urban status as the test variable. The app shows a preview of the metadata variable, so you can verify its values. You then indicate whether the metadata is continuous or categorical. In this example, rural versus urban is categorical. Based on that choice, the app recommends appropriate methods. For binary or multi-category groups, options include Wilcoxon test, t-test, Crespo-Wallace test, ANOVA, generalized linear models, logistic regression models, and linear mixed effects models. You can specify whether the data should be log transformed and normalized and how the results should be sorted, for example, by FDR. After clicking Run, the app shows a table of taxa ranked by their FDR-adjusted p-values with the smallest FDRs at the top. The table also shows which group each taxon is enriched in and the full change between groups. A histogram of p-values is provided as well. If you see an excess of small p-values below 0.05, that suggests real signals are present. If the p-values are nearly uniform, the differences may be largely random. You can also fit more complex models. For example, with linear mixed effects models, you can adjust for covariates such as age, sex, or batch, and you can specify random effects when there are repeated samples from the same participant. For logistic regression, you can choose the metadata as the outcome. For example, you can model rural versus urban status as the response and use taxon abundances as predictors to see which taxa are associated with living in rural or urban areas. When you want to test associations with continuous metadata, such as age or dietary intake, you can switch the metadata type to continuous. The app then offers correlation methods such as Pearson, Spearman, and Kendall, as well as linear models, logistic regression, and mixed effects models where appropriate. In this example, we select age and start with Spearman correlations. The app returns which taxa are positively or negatively correlated with age. We can then rerun the analysis adjusting for rural versus urban status, so the results show associations with age after accounting for location. Next is the tree plot module. For clarity, we will return to rural versus urban status as the main comparison, but you can also use correlation results. We start by running the tree plot. The parameters control how much the tree is pruned. If the tree is too dense, you can reduce the number of nodes shown to make the visualization clearer. You can choose which domain to plot, such as bacteria, archaea, or fungi, depending on your dataset. In the tree visualization, the rings from the center outward correspond to taxonomic levels. For example, the innermost ring might be phylum, followed by class, order, family, genus, and species. Nodes that are significantly different between groups are highlighted. For example, nodes in red may represent taxa enriched in urban samples, while nodes in blue may represent taxa enriched in rural samples. Larger nodes indicate higher relative abundance. This tree plot provides a nice overview of where the significant taxa lie on the taxonomic tree and how they're distributed across different levels. If you want to inspect differences for specific taxa in detail, you can use the box clot module. This clot is similar to what appears in the AI repair, but with more control. You can change the FDR cutoff used to select taxa and reorder groups. One useful feature is the ability to search for specific taxa. For example, if you want to see bifidobacteria, you can type part of the name, such as bifido, and run the plot. You may need to relax the FDR cutoff if the taxon is not significant. The app then shows the box plot for bifidobacteria in rural and urban samples. Similarly, in search for lacto to quickly plot lactobacillus without scrolling through all taxa. This makes it easy to focus on taxa of particular biological interest. The correlation plot module focuses on continuous metadata. To use this module, you first run the statistical test with a continuous variable in the previous step. For example, you run Spearman correlations with age. Those results are then passed to the correlation plot module. The correlation plots show which taxa are significantly correlated with the continuous variable. You can again select specific taxa, such as bifidobacteria, and adjust the FDR cutoff. The plot displays the relationship between abundance and the metadata, colored by another variable if desired. For example, you can color points by sex to see whether patterns differ between males and females. Final module is the p-value versus p-value plot. This is a powerful way to compare differential taxa between two related analyses. For example, you can compare rural versus urban differences in male participants versus rural versus urban differences in female participants. This module uses two sets of p-values. You can either upload existing p-value files generated elsewhere, or you can generate new p-values directly in PlotMicrobiome. In this demo, we will generate new p-values for both groups. For dataset 1, we select gender as the stratification variable and choose male. We set prevalence and abundance cutoffs, choose rural versus urban status as the test variable, and run a Wilcoxon test. This gives us p-values for rural versus urban differences in male participants. For dataset 2, we repeat the process but select female as the stratification variable. This produces p-values for rural versus urban differences among female participants. Finally, we run the p-value correlation plot. We tell the app whether to account for the direction of change, that is, whether the taxa is higher in rural or higher in urban samples. We can also customize point colors and label options and set cutoff thresholds for highlighting points on the resulting plot. The x-axis shows signed negative log 10 p-values for dataset 1, and the y-axis shows signed negative log 10 p-values for dataset 2. Points toward the top and right represent taxa that are significantly higher in urban samples in both males and females. Points toward the bottom and left represent taxa that are higher in rural samples in both groups. For example, Bacteroides may be higher in urban samples for both males and females, while Collinsella may be higher in rural samples for both groups. You may also see taxa that differ only in one group, such as taxa that are significant in males but not in females. This visualization helps you quickly see which differential taxa are consistent across subgroups and which ones differ. This concludes the PlotMicrobiome demo. We have walked through data input, normalization, and metadata setup, the AI-powered analysis report, ordination and diversity analyses, composition plots, filtering, a wide range of statistical models, tree-based visualizations, box plots, correlation plots, and p-value comparison plots. PlotMicrobiome is designed to make microbiome data analysis more accessible while still supporting rigorous and flexible statistical workflows. Thank you for your time. If you have questions or suggestions, you are welcome to contact me by email or submit feedback through the app. Disclaimer, this is AI voice transformed from a human's demo.